Just over a year ago, a group from Colorado State University came and visited us at the Land Institute. We've known of the Natural Resources Ecology Lab, group of ecologists at CSU for a long time, very prominent group of ecologists, and it was a pleasure to start to interact. And Francesca, we tried to get some funding. We didn't get that funding, but that didn't hold Francesca back from suggesting to her PhD student that she actually use some of our research plots in a project that will relate to what she's going to be talking about, I have a feeling, in a few minutes. So yeah, so Francesca is a professor in soil and crop sciences and a senior scientist at the Natural Resource Ecology Lab at Colorado State University, and there's a great deal of interest in decomposition and how soil organic matter is formed and is stabilized, and looking forward to a great talk. So Francesca.
Thank you. Thank you, Tim. It's really great to be here. It's I guess I've always studied perennials. I study ecosoils from the Arctic to the tropical forest. So I worked more in perennial ecosystem, including the Kansas prairie, than in agricultural systems. But I got to this group thanks to actually an interest on how to solve issues in conventional agriculture. And so let's start from what we have started from for a lot of these talks, and is that today we are living in this world where there is an increasing number of people that demand energy and consume energy, water, and food, and we have to address all of that in the context of climate change. And that may be a little bit partisan, but I believe that the soil is very much at the nexus of all these issues that we are dealing with, in particularly because the majority of our food is grown on soils. The soils regulate climate, regulate water cycles, and now we are looking more and more at soils also for bioenergy production. But in general, soils provide a lot of what we know as the ecosystem services. And actually, I'll be even more partisan by saying that within the soil, the thing that really matters and that really produce all these services is the soil organic matter content. Is the soil organic matter that maintain the huge, beautiful biodiversity that is in soils that, Jim gave a great talk and is very important on controlling also all the functioning of the soil and productivity. Of course, is soil organic matter with its recycling that provide the fertility to support, that should provide the fertility to support food production, and of course is through soil organic matter the water get retained in soils and purified and reformed in the aquifers. But of course, soil organic matter is extremely vulnerable, and the biggest impact of conventional land use for grazing and cropping through the 12,000 years that we have done it on the planet has been estimated to have costed about 120 petagram of carbon, with the majority of that being gone into the atmosphere. So this is the price we paid, and this is a very important graph to look at and think, okay, first of all, we can't move anymore in that direction, and secondly, I think the good side of it is that there is an opportunity we have to bring it back. And so there are a ton of soils in the world that have the potential to store that much carbon, and we have to think about how to do that, also to support, of course, agriculture in the future. And so we are now looking at soils as an opportunity to provide solutions, the natural-based solutions, soil-based solutions. And one thing I want to make clear, because there is a lot of fight back actually from soil scientists. They're saying, you know, if you push for carbon sequestration, it takes the attention away from the fight on fossil fuel burning. So the point here, of course, is that the first and, you know, undoubtful thing that we need to do is to transition away from fossil fuel consumption. But that is actually not even enough. And on top of that, we also have to sequester carbon into the soil. So we are looking at soils as a way to remove carbon from the atmosphere and sequester carbon. But that actually might conflict with the fact that we also want that carbon to recycle so that to mineralize so they can produce nitrogen for plant growth and release for the excessive use of fertilizer. But that process moves carbon back into the atmosphere. So in a way, right now we are looking at soil as the foundation to solve two problems which might present an issue which Henry Janzen, who's one of the most intellectual soil scientists I have the pleasure to know, proposed it as the soil carbon dilemma. Are we going to store it, or are we going to use it for natural fertility of soils? That dilemma presented itself, in my opinion, because we were looking at soils from this standpoint. The standpoint of humification. So you have soil organic matter formed from what didn't decompose and accumulated. And so the formation of organic matter has been always seen conventionally as being opposed to the transformation and the decomposition process that would consume that organic matter. This humification theory is now been totally dismissed by the soil biogeoscientists and ecologists. There is still a community of soil scientists that, you know, are in the humic society and still defend that view. But a lot of us have moved to this vision of how soil organic matter is formed, and this vision is radically different from the standpoint that actually the stable form of organic matter happen after microbial decomposition or as part of microbial decomposition. So there isn't necessarily an antithesis between microbial transformation of plant debris and formation of organic matter. And the reason bioorganic matter stay in the soil is not because it's biochemical recalcitrant and not decomposed, but because there are some other further inhibition of its transformation due to its association to minerals, due possibly to climate constraints, or to other constraints dependent on the physical structure, if it's, for example, a deep soil, or if it's in aggregate, and so forth. So on this vision, I proposed a framework that linked together the decomposition of organic matter with the formation of the decomposition of plant debris with the formation of stabilized organic matter on minerals. And so I'll drive you to what I call the MEMS framework, the microbial efficiency mineral stabilization framework. Basically, according to this framework, depending on your plant input, you might have fast or slow mineralization, but this fast or slow is matter of possibly years. And what I'm claiming is that those years actually in the scheme of things are not that important. What is more important is the efficiency of the process. And so as we were talking yesterday about the fact that us humans actually like to burn most of what we eat and go to the gym and so forth, in this case, it's how much of what microbes eat produce microbial biomass and microbial products versus how much is respired off as CO2. And so what I'm claiming is that actually the most labile plant material that can be an alfalfa leaves or you know, a root, a fine root or so forth, they turn over fast, but actually are very efficiently consumed by the microbes, in particular because they are more nutritious, they have a lower carbon to nitrogen ratio, and so the microbe retain them more. As well as they have less of the structure, the lignin structures that have a very high energetic requirements for microbes to build, and so require a lot of investment. So what I'm saying is that after the microbial substrate use efficiency filter, if you forget about time, you start having, yes, this might happen in a month and this might happen in 10 years, but at the end there will be more carbon passed from this process than this process. And we now know that the carbon that is stabilized on the mineral surfaces, which is the one that stays in soils for centuries to millennia, is mostly determined by microbial transformation, or at least is labile. It could also be just DOC that comes off and immediately stuck on minerals. And so the point here is that you actually might have, on the longer term, more carbon formed from the decomposition of labile structure than from the decomposition of plant structures. But there is another story to this. And that's if you get one thing from this talk, I would like you to consider that organic matter is not one thing. Organic matter is actually highly complex, and one of the failures, I think, of us soil organic matter scientists is that we go into the minutiae of that complexity to the point that people cannot follow us. And so my goal in the last year or so has become to say, okay, in this complexity, what are the really two major pools that we need to separate because they are very different? They are produced differently, they are chemically differently, and they play a different role in soils. And so those two pools are actually the mineral-associated organic matter, and I told you how that pool got formed. And then there is another very, very important pool, which is the particulate organic matter. That pool is mostly derived by the plant structural as well as the structural of the fungi, for example. So as the recalcitrant plant debris decompose, microbes, in particularly fungi, grow into it, making that particulate organic matter that then ferment and get into the soil. That is also a very important part. This part decomposes faster, but also can make a significant component of the soils. And I have analyzed over 10,000, about 10,000, so actually 20,000 soils. What is missing from these are all the agricultural soils that we are about to get the data now. These are all soils that were collected across Europe from the Lucas database. And they are only the 0 to 20 centimeters. What we did was to fractionate in MAOM and POM about 400 of them, and then do a random forest to predict across the entire database. And that's the prediction. In blue, you have the grasslands. Grasslands in Europe are a bit of everything. So they are not necessarily natural grassland. The majority of them are highly managed, highly fertilized. But in this talk, they are just defined as grassland, blue. The red are the forests, and the darker reds are the conifers, and the pale red are the broad-leaved forest. And so what you can see, first of all, is the MAOM and POM are these very contrasting pools, with the MAOM basically reaching a saturation level, mostly imposed, I think, from the amount of minerals there, but also from the processing. You can't accumulate MAOM forever, whereas coniferous forests, in particularly up in Scandinavia, they have a ton of POM, and that's where they really accumulate. And this is the mineral soil. This is not the organic layer. So the point I'm making here is that now we have two pools that maybe function differently, from which we can use one for storage and one for mineral production, or anyway, we have more leverage to discuss how to do this, both carbon sequestration and provision of fertility. The other thing to remember, and that's actually a paper that came from a 3-year study in the Kansa Prairie, studying the decomposition of big blue stem. What we found out was that during the early stage of decomposition, this is time, here we are at 3 years, and here we are at about 6 months. At the beginning of the process, when the litter lose the more labile substances, DOC, but also non-lignin encrusted celluloses and other polymers, what we see is a very fast and microbial efficiency. We see a lot of that carbon stuck in the polylipid fatty acid of microbes at that stage. And then we see it forming this mineral-associated organic matter. Later in the process, the litter residue is mostly made of this anhydrolyzable fraction, which contain lignin, but we did mid-infrared and contain also a lot of amides and fungal material. That is fragmented and moves into the soil and form this particulate fraction. The beauty of these two fractions is that they are physically distinct. This is finer and heavier, this is coarser and lighter. And so we have actually a good way to separate them and measure them. The other point to the story, and Jim has said all about it, which was great, is that in order for the soil to function and so we have made one postulate, and is that soil organic matter actually forms thanks to microbial transformation. Microbial transformation don't have to be inhibited to get organic matter formation. And the second thing is that these microbial communities, actually, the more they are connected to the roots and the plant, the better they function for also this transformation and soil organic matter stabilization. And there is a lot of discussion now about the rhizosphere community and the bulk soil community, and how it's important that they connect in order to get both a carbon and a nitrogen efficient transformation inside the soil. So with that in mind, a couple of years back, I wrote a USDA proposal, and you heard, I'm actually Italian, but I work at Colorado State University, and our big deal is wheat, dry wheat, and wheat fallow is a very, very common practice around there, which, of course, has all the huge issue of conventional agriculture. Even with a season of fallow, you have terrible erosion and so forth. So the point I was making here, saying that we could possibly address this soil carbon dilemma in basically putting into the soil continuous inputs, but also diversified inputs. So you need to have the carbon input, but you have it to have in a way that can both form MAOM and POM, and can both have the nitrogen to provide the fertility. And so there were other studies before that said that if you move from a wheat fallow into a continuous rotation of grains, you increase the carbon. But my point was that that you still don't provide the fertility, and you still don't provide the microbial connectivity. And to do that, you needed to have a legume. And so I set off with that experiment. I got funding from the USDA, and we did a very big campaign both at the Akron station in Colorado, where there is this rotation in an experimental farm, as well as sampling a ton of farms all the way up to Nebraska Unfortunately, in Colorado, there are not many farmers that put pea into the rotation, but in Nebraska, it's becoming more common. So we farmed a bunch, we cored down to a meter a bunch of in-farms as well as experimental station, and the work is mostly done by Loya van der Poel, who's one of my PhD students. And the data are coming, but to be honest, are not that revealing. We are seeing something, but is nothing particularly exciting. And that, at that time, we went and visited the Land Institute, and Tim, drove in that car very nicely around all his plot. And as I was seeing those plots for the first time, I said, hold on a second. I mean, those make much more sense for my question than the wheat fallow rotation. And so we have, I had another point before that. I'm jumping ahead to my story. But the reason why I got to that point was because what I was thinking is that it might not be enough this adding another culture that still is not continuous, and also the fact that the input was coming from above ground versus below ground in a perennials. And that is some other work we, for example, did in another kind of crop, which is actually poplar plantation in Belgium, where we looked at the contribution of the above ground versus the roots into the formation of organic matter. And what we found was that while in this system the majority of the carbon come from the above ground, the below ground was much more efficient at forming organic matter. And so again, it's not that important just how much you put in. It's important if it comes from the root or if it comes from above ground. And I think it's important whether it's continuous or pulsed, because if you have that pulsing thing, I think you still disrupt that plant microbial connectivity that you can preserve if the roots are perennials in the soil. And the other important things about whether you have the input coming from above or below ground has to do with this idea that we are now very pointing to in soil science, which is the point of entry. And the thing is, if you remember my framework, that you need microbial processing and mineral stabilization to create that thing, here the question is, which of the two is more important, and do they differentially apply if you come from the roots or come from above ground? And so the point is that roots actually have a very different way of dying and decomposing, because they are still attached, they come from a living environment, very rich, as we have heard just before, in these microbes. And so as the root tissue die, there are already there a very active microbial community that can transform it and produce those microbial products that then might form this efficient mineral stabilized organic matter. In the, of course, that happens when you have a matrix capacity. In sandy soils, that cannot happen. And so in sandy soil, you are actually better off trying to put in some structural component. When you get this from above ground, actually the above ground plant material enter in what we call a microbial desert. The bulk soil has very little microbes compared to the rhizosphere. And so we now think that it's very important if the carbon comes from the roots, from the live roots, versus from the above ground. And that, I think, the core issue in perennials is that you have the carbon coming in from live roots. And that's what excites me about trying to do this story. But there is a but to that. And the but is that input doesn't necessarily result into carbon formation. And then I make the example when I teach priming to my student is like when you go for an appetizer, you kind of stimulate your metabolism, and then you consume more afterwards. And what happened here, actually, the reason why, if you give a lot of fresh carbon to microbes, they might go and actively decompose the native organic matter to get at the nitrogen. So that's another point where the nitrogen becomes very important, and why it's not enough to give carbon. You also need to give a balanced carbon, and that's where the diversity come about together with the continuous inputs. And so if you have more nitrogen, you actually don't need, the microbe can access the nitrogen and don't need to prime the native organic carbon to get at the nitrogen. And we studied this process across a large number of soils and actually found that it's a very universal phenomenon. And in grassland, I'm sorry it's so little, in grassland actually was a pretty high carbon priming if you had, for example, celluloses. And so the idea here is that, yeah, you can get more if you make continuous, but you do need those legumes. You do need that nitrogen input in order for not resulting in priming and in order to do the actual carbon sequestration. So with this hypothesis in mind, and I'm sorry, I couldn't find a different picture. So those are the two pictures, but we will be working at 3 different plots. With this idea in mind, and thanks to team interest in our question and hospitality, actually next week, my student is going there to set an experiment to test those questions and understand how either just Kernza with or without nitrogen fertilization, or a Kernza alfalfa with the natural fixation would affect this carbon and nitrogen recycling. The way in which we ask these questions, because we need to trace carbon and nitrogen from the residue into the soil, is to use isotopes tracing. And we do that at Colorado State. We have a labeling facility, a labeling chamber that I built as I arrived there, where we produce plant material. This is actually the big blue stem when we did the experiment at the Kernza Prairie. We produce this labeled plant material that then we use in all kinds of experiments. The basic principle is to incubate the litter in a way that is very, very similar to the native situation. One thing that I've totally moved away from is, for example, using litter bags for decomposition. That's a technique that I don't think is good in looking at carbon dynamics. But for example, we also have a lysimeter to collect DOC, and then we have done some work in the Arctic, for example. I am interested in fire, and so I also burn that material, produce pyrogenic carbon, and do similar experiment. More recently, we have been interested in mycorrhizal, and so we grow labeled mycorrhizal to then do field experiment. And we are going more and more, now that we study roots, we have done work all the way down to 1 meter, where we incubate that material into meters. The problem with my thing is that I still just incubate residue, so I don't have a way to do that live root that becomes detritus, and that's why I was very interested in the Danish root towers. With that, we do a bunch of different things. We can just look at the soil, but as I told you, I'm a proponent of looking at different fractions. We can look at gas fluxes, we can look at microbial proxies, and then we go all the way very geeky. We do some nanosims work where we actually see where those carbon and nitrogen are. We have a lot of different experiments in which we do this all across the world, and I was saying more of that into perennial than agricultural system, but a lot in agriculture. And that's the design that we are going to use in at the Land Institute, where we'll be doing both Kernza and alfalfa on above ground or below ground in all those plots. And it was very nice to grow actually the Kernza and the alfalfa in our labeling chamber. Just briefly, we have done that in many different things. I know that there is a community of people interested in sorghum. We have done with sorghum down to 1 meter, and you see the blue is the decomposition of the detritus, and then the lighter, the darker green, and then the lighter green is the soil organic matter formation, both for carbon and nitrogen, and we have more formation for roots. We can go all the way into seeing how that happened inside aggregate, outside aggregate, and so forth. So it's a very powerful approach. And then the nice thing is that we have now built a model that uses those fractions, and so we can model these results and see how they work. So in conclusion, I think that polyculture and perennial grains and legume may allow for this, for solving the soil carbon dilemma, may allow for formation of organic carbon. That's what happened in nature. One thing that this conference has been very different from any other conference I've been in. And so I like to conclude with something that I think, but I've never had the courage to tell anybody. So I am a biogeochemist and an ecologist. And I look at the world as, we are basically, in general, animals don't actually serve much in the big scheme of biogeochemistry. They consume energy, they dissipate energy and consume resources. And thanks to the fact that autotrophs produce so much, we can exist. So that's and the reason why we have been, we heard that yesterday by Tim, we have been so successful because we find the way to be the best consumer and the biggest dissipators of energy, and that has what given us the success we have become in at the expenses of our planet. And so I was thinking before getting to this community that is so focused in agriculture, that actually ecological intensification is something that we should think of in our socioeconomic system. Because the basic principle of competition and the fact that production is the goal of our society are not intrinsic in nature. Those are not the goals that assure ecosystem to maintain. They might be goals that assure the short-term success of an individual or a population, but they never allow the success of an ecosystem, as we have heard in the case of the mycorrhizal. And it's actually the higher number of positive relationships. It's not a case that you have mycorrhizal later in succession. That's an evolution. As well as multiple and diversified process pathways. You need to have diverse microbes that do the same thing, diverse plants that all do photosynthesis to make sure that they don't get affected by a disturbance. And we are going in a world that is highly disturbed. And so I think that our goal should be the resilience to disturbance more than just the production. And so that's what I think. And again, I never had the courage to tell anybody. And where I start actually in schools. I go to elementary schools and I do the food web game, and I teach the kids that, if one group die, the entire web collapse. And so that's something that I'm very interested with. And with that, sorry if I took too much time, and thank you.
Well, thank you, Francesca. That was a fabulous talk, so informative for even those of us that think about these things a lot. And we are nearing the end of our time in this room. I think we can take 2 questions. and then we'll have to have subsequent interactions over meals and at the Inferno later tonight. But Ed, is there a question? Oh.
Okay.
Okay, Leonard.
What is Shell doing up there?
Shell is actually, Shell has a very high, they just hired one of my postdocs. They have a very active group in research and development, and they are strong in natural solutions. So they are funding a lot of work on carbon sequestration, whether it's, I think they are not stupid. They are looking into the future. And they don't want to lose their money, so they want to have the next thing to jump in. So they are getting prepared. And if you wonder, McDonald's, we have a project on sustainable grazing, and actually McDonald's has given about 4.5 million to that project.
Thank you very much. That was excellent. I'm essentially a cropping system designer as an agronomist, and I have learned about this paradigm of soil organic matter accumulation a number of years ago, and it has made all the difference in how I think about designing systems. And many of my soil science colleagues are with you. Many of them are still in the dark ages. Unfortunately, it's very difficult. So
the problem is that we teach our kids in the same department. We teach differently.
Yeah. My question is, what would be your elevator speech to cropping system designers who are thinking about cover crops and polyculture and multiple species, to encourage them to think about designing for this outcome?
Yeah, again, I think that the key is inputs and diverse inputs. So there's no way you can do organic matter without the inputs. But the inputs are not enough. They have to be balanced. Carbon and nitrogen have to be balanced to feed the entire microbial thing. And so I think that that's the perennial. But managing rotation for high and diverse carbon input, possibly along the depth.
All right, well, we're going to have to close it there, but let's thank Francesca again.